Now there's a new person doing it this year, and basically she's like, her brain really works good, so I just sort of. It's coming. It'll be your next year. It'll be your first year. No, it should be a next year.
See, I'm
So I
JAK-STAT here we go. Look at Mike, he's where he's just a businessman. Just got here on the bus. Reading his paper.
Got to get his fourth page. Otherwise, I won't read.
Oh, okay.
The Mariners actually won last night.
I know, they're actually looking like they're good.
There are a lot of people there.
That's what you have to do.
We're looking at that script here. Oh, you got the information? Yeah. It's an intriguing one. It's not very expensive, really. Not bad. I don't think it's a Prince Charles.
The price, like I said, they cut them in half immediately.
Yeah.
It wasn't the price at all.
Well, that may be true. And, you know, we signed up on top of it 4 or 5 years. So that might make a difference, too. What is that looking like for the cheapest Autosomal stuff, they say? Yeah, so the whole basically the same price. That's good. I thought maybe it'd go up by now. We got a spreadsheet on it.
I've been told if you wait to the very end, the sale will sold at the price.
Oh, you can take that.
Yeah, will I take that? Check, I really want to go.
Well, we had the same deal. We got a trip we're signing up for a couple of years. I haven't want to do it this year, but I waited because just trying to make it. It's just a Scandinavian. This would be a place in between. And by the time we got ready to sign up, they raised the price to plus it was also it's great. We need to sign up 3 years in advance to get it.
They are.
Viking River Cruise it's the new boat. In fact, the new boat is the first one this year. But it looks like a very good trip. Actually, quite frankly, we got 10 results signed up. It was just a great trip. All tours and food. I know. Long time. So anyhow, the rips are fun. How's it going? Yeah. Sorry, I haven't let you know slides yet. I'll do it today.
Actually, I found stay home. Oh, how'd the talk go?
Really good. What are we doing? Feedback. Yeah, so we got a very useful.
That's awesome. You're not clear this time. Yeah. You got a console to do a pre-pen animation.
Okay, I can do it.
I'm at children's all day.
Yeah, just send me.
I just don't want to.
I can pitch that for you.
Thank you.
Back.
two, I'd like to have all the fellows here if possible. If everyone needs to know this, the rest of us practice and forget it immediately. You got to take a test of it. Or if you even understood it in the first place. Not only that, I even forget the topic. I don't even know we did.
Is there a recording somewhere?
He just kept starting most of it.
It's amazing.
Lisa's the presenter today. For those who signed up for CME, the word of the day is, as you can see, interferon. And she's got a pretty heavy-duty topic here, but something that, like it or not, I think we need to know. JAK-STAT in allergy, immunodeficiency, so should be a very interesting topic.
Thank you. Okay. I hope this will be an interesting topic. As Dr. Altman mentioned, this is about JAK-STAT and immunodeficiency. It will be relatively involved, so I hope you guys can all follow along. I'm by no means an expert in the topic, so those of you that are an expert, if you want to step in at any point, feel free. So basically, I'm going to give a bit of an overview about the different JAKs and STATs, start with some definitions and review of the cell signaling pathway, and then go into some specific mutations in each of the JAKs and STATs and what kinds of clinical phenotypes you'll see with regards to immunodeficiencies related to those mutations. I have no disclosures. Okay, so JAK-STAT signaling. Basically, there are a bunch of different cytokines that use this pathway, and there are type 1 and type 2 cytokines that include things like interferons, interleukins, colony-stimulating factors, and they basically all share the same signal transduction pathway, which is the JAK-STAT pathway. So JAKs, it stands for Janus kinases. These are tyrosine kinases that selectively associate with the cytoplasmic portion of cytokine receptors. When they interact with the cytokine receptors, they then phosphorylate the receptors when they become activated by signaling. STATs are signal transducer and activators of transcriptions. These are DNA binding proteins that become phosphorylated themselves. When the cytokine receptor is phosphorylated, it then attracts the STATs to come. They become phosphorylated as well, and then they're able to dimerize in partners, tubes, and then they are able to translocate into the nucleus, and by doing so, that is what helps regulate gene transcription. So here's a wonderful schematic. As you can see, the cytokines come in, they bind the receptors. On the cytoplasmic domain of the receptor here, you see the JAKs associated with the receptor. They cause phosphorylation of these tyrosine residues. And then the STAT proteins can come in. They have a portion in their SH2 domain where they also have tyrosine residues that can then become phosphorylated. Once the STATs are phosphorylated, they can dimerize, and then they go inside the nucleus and help regulate gene transcription. So the JAK-STAT pathway, knowledge of it is not that old. In the last 50 or so years is when things kind of really got discovered. Back in 57, Isaacs and Lindenmann were the ones who first discovered interferon, which kind of spurred on further discoveries of the signaling pathway. And what they found was in influenza virus-infected chick embryos, they were able to discover this protein that seemed to help prevent infection prior to the production of antibodies. It seemed to be part of the innate immune system, and this protein that interfered with the virulence of viruses, they called interferon. And so once interferon was discovered, there was a lot of interest in kind of figuring out more about this pathway and how interferon mediated the effects that it had in terms of helping fight off viral infections. This is just a schematic that kind of shows you over the years the different things that were discovered with regards to the interferon signaling pathway. It wasn't until the late 80s to 90s that people started to discover the JAK kinases and the STAT proteins as part of this pathway. The first one of the JAKs that was identified was actually tyrosine kinase 2. It was a tyrosine kinase, so they labeled it as such. There's other JAKs as well. And then throughout the 90s is when more of the STAT proteins were identified. So this is just a summary slide of the different family members in the JAK-STAT pathway. There are 4 total JAKs that have been identified, and this is what the protein structure looks like. STATs, there are 7 total. Again, you can see the tyrosine residue here with the SH2 domain, which indicated gets phosphorylated when activated in order to allow dimerization. So we're going to go ahead.
Laid out there makes it look like they're on a chromosome. Are they just adjacent to each other like that, or is that just a
No, this is just to show you kind of what they look like in terms of the different domains in each of the JAK and the STAT, but they're not next to each other. Okay, so JAKs, this is just kind of a broad overview again. There are 4 total. JAK1 and JAK2 actually have very broad functions. Their roles range from helping with things like the immune host defense, but also they're involved in things such as hematopoiesis, growth, neurodevelopment, whereas JAK3 and tyrosine kinase 2 are more important for immune responses. And actually, JAK1 and JAK2, if you have mutations in those pathways, in mice, JAK1 and JAK2 mutations are known to be lethal. JAK3 and tyrosine kinase 2, there are human mutations described, and we'll go over those in more detail later. Again, this just kind of summarizes JAK1 and JAK2, saying how mutations in these pathways have been found to be fairly lethal, which is why you don't really see them in humans. But JAK3 and tyrosine kinase 2, JAK3 defective signaling through the cytokines IL-2, 4, 7, 9, and 15, and also 21, which are the cytokines that signal through the common gamma chain. When you have defective signaling through that pathway, JAK3 is affected. This can happen either by mutations in the common gamma chain receptor or mutations in JAK3. And basically the resulting phenotype you get there is SCID, which we'll talk about in more detail as well. Tyrosine kinase 2, if you have issues with your mutations in that, you will have decreased response to signaling through interferon alpha, beta, and IL-12, and this primarily will lead to issues fighting off viral infections. Going over the stats, so there's 7 total. Stat 1, 2, 4, and 6, they basically have various roles in host defense and immunoregulation. You may recall that Stat 6 is important for activating the Th2 pathway. Stat 4 is more involved in the Th1 response. And then other ones that I'll go into more detail about include Stat 3 and Stat 5b. Stat 3 has a broad role. Complete deficiency in mice is lethal, but we're going to talk about specific autosomal dominant Stat 3 mutation later. And then with regards to Stat 5a and b, they're also obviously important in the immune response, but they're also involved in prolactin and growth hormone signaling, which will become important in one of the mutations that we talk about with regards to Stat 5b later. This just kind of goes over the different things you'll see with regards to defects in certain stats, and we kind of touched on this a little bit already. But primarily with Stat 1 and 2, you're seeing decreased responses to viral infections. With varying degrees of severity, Stat 1 mutations tend to have greater degrees of severity in terms of being able to fight off viruses than Stat 2, which we'll talk about in more detail as well. And I'm just going to move on to the next slide. Okay, so this is, again, an overview that just kind of shows you the different cytokines that are associated with certain JAKs and the certain stats that they're associated with. So here again, we have cytokines that share the common gamma chain and the JAK kinases that are associated with them, the stats associated with them, so you can kind of see them in their groupings there. Down here you have interferons and the JAKs and stats that they are associated with. So I'm going to get into talking more about specific JAK and STAT mutations and the types of resulting immunodeficiencies and clinical phenotypes you see based on those. This is basically a slide that's showing when you have mutations in certain JAKs and STATs, the resulting things that you'll see from them. So you see a variety of different cytokines and growth hormone and other factors that are signaling here. Basically, what we're going to focus on are the parts that result in some kind of immune deficiency or issues with allergic disease. Obviously, with some of these, you can have gain-of-function mutations that will cause things such as cancer and other myeloproliferative disorders and hematologic disorders. What we're going to be focusing on here is mostly the immune deficiencies and difficulties with the immune system regulation. So as you can see, most cytokines interact with a variety of different JAKs, and there's certain JAKs that are associated with multiple different cytokines. JAK3 associates only with the common gamma chain subunit, and so that becomes important with regards to disease you see there. The tyrosine kinase 2, again, causes immune deficiency. We talked about how STAT1 mutations will predispose you to viral infections, as will a STAT2. We are also going to talk about STAT1 and its gain-of-function mutation and how that leads to a more distinct phenotype. And then we'll also touch on STAT5B, a kind of unique phenotype you see with mutations there as well. This is a summary slide. We've actually seen this before from Dr. Torgerson's talk, but again, just kind of highlighting when you have loss of function of certain STAT and JAK mutations, the kind of phenotypes that you see. Again, differences with regards to the type of immune deficiency. You're going to see STAT1 and STAT2 basically deficiencies fighting off viral infections, and then more distinct phenotypes with STAT3 and STAT5B and JAK3 deficiencies. The gain-of-function disease, I'll talk briefly about STAT1, but we're not really going to go into the others as they're not as important with regards to the immune system. Okay, so to start with the JAKs and the mutations that I'm going to discuss there. Again, this slide is just showing you cytokines and which ones associated with which JAKs when they are signaling. As you can see, the JAK kinases are involved in signaling with a variety of different cytokines. So for JAK3, sorry.
In normal biology, how does this thing sort itself out? I mean, how do you observe specificity? If so many cytokines can use various JAKs, how does it figure itself out to have normal function? How do you get a specific cytokine to work with a specific JAK with a specific STAT to get the end function you want?
Yeah.
I mean, part of it is there are different receptors and different receptors on different cells.
Right.
So even though, all of these use JAK, there's something upstream of that, which is the receptor. A different receptor for each of these cytokines.
So it's tissue specific?
Well, no, even beyond that, it's receptor specific, and tissue specific. You have both issues leading to something. But
so the organism figured it out for itself.
Okay, so we're going to talk about the specific different mutations and the resulting phenotypes that you see. We talked about this a little bit already, but with regards to JAK3, so common gamma chain, the common gamma chain receptor for IL-2, which is shared between multiple different cytokines, the signaling of that specific chain depends on JAK3. So there was described X-linked forms of SCID that involve the gamma chain mutation, and these patients always have a T-negative, B-positive, NK-negative SCID type phenotype. It was discovered then, however, that there were some patients who had this phenotype that did not have the X-linked mutation, such as females, and they were found to have a JAK3 mutation. This is an autosomal recessive mode of inheritance, but they basically have the same type of clinical phenotype with regards to SCID, which makes sense if you think that the common gamma chain and JAK3 signal through the same pathway. This is not the most common form of SCID. It's relatively rare, approximately 6% of all cases. And again, this is just showing the different cytokines that use the common gamma chain receptors. Obviously, IL-2, you see the common gamma chain also for 4, 7, 9, 15, and 21. And then again, JAK3 associates with this specific subunit fairly specifically. So if you have a defect in the common gamma chain, this can result in SCID, but also a mutation in JAK3 with normal common gamma chain expression can still cause the same defect. Certain receptors by themselves, if you take out the IL-7 receptor, for instance, you have IL-7 receptor deficiency can cause another type of SCID. They feel that based on the phenotype you see there, which is a T negative, NK positive, B positive, rather than NK negative, as you see with JAK3. Yes.
Now, if you have these cells with this deficiency and you replace the JAK's protein, does that correct the deficiency? Or is the disease just associated with a lack of function?
So the if you're talking about JAK3 mutation leading
Yeah.
to SCID, if you were able to
Just give them
replace,
JAK3.
then
The cells work fine
essentially
after that.
It should. Because what you're seeing with that mutation, again, it's an autosomal recessive, is a loss of function of the JAK3. So if the JAK3 were replaced and you were able to give them a functional version of JAK3, then in theory, yes, it should work.
So is it toxic enough that you couldn't give that to one of these kids? Just give them an infusion of JAK3 and see if
Well,
they get
it's an
better?
intracellular protein.
Yeah.
Part. So if you just give it IV or something, it's not going to get where it needs to go. It's not a.
It's not like
You can give them
blood
cells
cells
that
or
are
antibody.
So if you transfect a cell or something, you could do that.
Sure, no problem.
Yeah, easy.
Just
It's
ask.
easy to replace extracellular stuff, but it's hard to replace, membrane proteins or intracellular.
Okay. Good question. What they've shown, though, that is certain ones of these cytokines have different properties. So, for instance, as I was saying, if you were to knock out a portion of the IL-7 receptor, not the common gamma chain, but the alpha subunit, for instance, that results in a different type of SCID phenotype, which is predominantly T-cell deficient, but you do have NK cells. Whereas if you knock out JAK3 or the common gamma chain, you see a different phenotype. So they feel that IL-7 is specifically responsible, or in large part responsible for that severe T-cell deficiency phenotype, whereas different signaling through different cytokines and their subunits is responsible for the more NK deficient. So the whole picture that you see with common gamma chain and JAK3 mutations varies a bit than if you were to just knock out a specific receptor, such as the IL-7 receptor alpha subunit. This is a case study that also looked at kind of a variable phenotype for patients with JAK3 deficiency. This was one family that was just described. They had 2 children that were actually compound heterozygotes that had some variable expression of JAK3 and kind of variable clinical phenotypes. They did not have a SCID picture. They still had some expression of JAK3 based on the mutations that they inherited from their parents. They were a little bit different. The first patient had some degree of severity in terms of recurrent infections, had a lung infection that was unsure if it was a viral or bacterial infection that was relatively severe as a 1-year-old, did get severe varicella, and then had some occasional ear infections and urinary tract infections, and did have some recurrent warts, but was relatively healthy when this was published when they were 18 years old. However, their sibling seemed to have a more severe phenotype and developed recurrent issues with PCP and Giardia, and also had some other severe infections like varicella and bacterial pneumonias, etc. And ultimately, that patient ended up passing away. They actually tried to do a bone marrow transplant of that patient, but they died of sepsis a few days afterwards. But what was interesting is that they showed with this specific type of JAK3 mutation, which again was a compound heterozygous mutation, they were able to show that there was a little bit of activation of the STAT5 downstream from JAK3, even though there was really not very much in the way of phosphorylation or JAK3 expression from that particular individual. So this just kind of illustrates that although technically most people would think JAK3 mutation SCID, there are other types of mutations that are possible that can lead to different phenotypes. And this kind of ties back to the signaling that you see through JAK3, the cytokines that are dependent on it, and deficiencies in those areas, which could lead to predisposition to recurrent viral and other bacterial infections.
Explain how the first sibling was healthy by age 18. What corrected his defect?
Well, it's not necessarily the defect was corrected. It's more that the phenotype was kind of variable between the 2 siblings, which they thought may be due to them expressing different amounts. They were able to show that there was some JAK3 expression, although decreased from what they were seeing in the parents. They didn't actually look at the sibling who died in terms of what was being expressed there, but their presumption was that there were variable levels of expression of JAK3 between the 2, so that would result in more severe phenotype for the one sibling who ultimately was deceased, and the other child seemed to have some recurrent infections, but to a much milder extent. So tyrosine kinase 2, there have been a couple autosomal recessive mutations that have been reported in humans related to this. And this is important, again, for signaling of interferon alpha, beta, and some other various cytokines. The mutations that have been reported in humans, there was one that seemed to have hyper IgE syndrome type of phenotype associated with it, although the other didn't so much. What was common between the two is that they both had susceptibility to invasive mycobacterial infections as well as some other bacterial and viral infections. And this just shows how the tyrosine kinase 2 signals for various different cytokines, interleukins and the interferon, as I mentioned. It associates with different JAK kinases as well in signaling. This is a study that looked at one of those patients with the autosomal recessive mutation. This was the one that did have the hyper IgE syndrome type of phenotype. It was a Japanese individual. And basically what they showed here was that the individual really didn't have anything in the way of JAK1 expression when interferon alpha and beta were stimulated. Compared to the control, you see phosphorylation of JAK1, whereas the patient here did not have that. And that was to kind of look at supporting the idea that the tyrosine kinase 2 is needed to associate with JAK1 when signaling for interferon alpha and beta. We also saw that in response to IL-12, there was decreased phosphorylation of STAT4 in the patient compared to the control. And again, the STAT4 is important for promoting the Th1 response, so the increased susceptibility of this individual to certain viral infections is likely secondary to that. Also, what they found in this patient was that when they looked at the patient, which is in black here, compared to a healthy control, they took naive CD4 cells and looked at stimulating them and seeing what resulted. They basically saw a very skewed Th2 type of accelerated response in the patient, which had increased levels of IL-5 and IL-13 production. And they felt that that somewhat explained the more likely propensity this patient had with regards to atopic dermatitis and other signs that kind of went along with the hyper IgE syndrome. So that was an interesting feature that they noted in this patient. I'm going to move on and talk about the STATs now and certain mutations in the STAT pathway and the different immunodeficiencies we see with regards to that.
Before you move past JAK, are there any patients that have an excess of JAK function? Does that present with disease as well, or is that?
You can have, yes. We go back to the, sorry. So there are certain diseases that are associated with gain of function. For instance, we talked about how some of the hematologic disorders and myeloproliferative disease can be associated with JAK2. I didn't touch on this much in my talk because I wanted to focus on the immune deficiencies. But those JAKs and STATs that do have gain of function leading to more hematologic disorders or other disorders associated with autoimmunity, those are very attractive targets for things such as inhibitors of those. There is research being done on ways to inhibit JAKs in order to mediate that and not cause hematologic disorders and myeloproliferative syndromes and that kind of thing. Unfortunately, because most of the diseases that we're talking about involve a deficiency, can't exactly make up an inhibitor to target those. And it's, as Matt was alluding to, difficult to replace them as they are intracellular. But yes, it is possible to have gain-of-function mutations with regards to JAKs and STATs.
And on that point, there are now actually 4 FDA-approved JAK inhibitors, which are mostly 3 of them in humans, 1 for dog. And they're being used largely in cancer and rheumatology. But there was some very interesting talk about this at CIS for the use in allergy as well, because the one in dogs is specifically to treat allergy. It's not being shown here, but a lot of sort of Th2 inflammation can be mediated through this pathway, as well as inflammation that's seen in rheumatoid arthritis.
It just seems like if it's involved in so many different pathways, you'd think if there was an excess that you'd have a whole bunch of different disease entities instead of just chronic mucocutaneous candidiasis or polycythemia. It seems like it's a very focal endpoint for something that's as broad spectrum as this.
Yeah, I don't think it's been fully worked out. They found that this is a convenient pathway to target for some inflammatory conditions, but what determines who has excess inflammation through these pathways is not, there are no clear gain-of-function mutations. I'm sure there are other genetic things that affect it.
I was going to follow up on that. If you go back to that slide, is it understood why gain of function leads to chronic cutaneous? Why does it get to that phenotype?
Yeah, we'll talk about that in just a minute for the STAT1 gain of function mutation specifically. Great save. Thank you. So for STAT1, as we mentioned earlier, if you have deficiency of STAT1, whether it's autosomal dominant or autosomal recessive, you're going to have problems with interferon signaling, and that leads to mycobacterial and viral infections, particularly in the autosomal recessive form. In the autosomal dominant loss of function, you have more of a mycobacterial infection type of phenotype. You can have this autosomal dominant gain of function mutation, and what this causes is an exaggerated response to interferon gamma, and it leads to decreased IL-17 production, and we'll talk about how that happens in a little bit more detail. But the resulting clinical phenotype you see is this chronic mucocutaneous candidiasis. So what happens, or what they propose happens with the gain of function mutation that leads to the chronic mucocutaneous candidiasis is that it causes, mutations have been mostly identified in this coil domain of the protein, and what they find is that this impairs the dephosphorylation of STAT1 so that it's chronically in an activated state. It's not able to be dephosphorylated. And because of that, what they see is a stronger cellular response to the signaling of the interferon alpha, beta, and gamma. This leads to a skewed inhibition of IL-17 producing cells, and when those are inhibited, they see lower levels of IL-17, obviously, and that's what they propose is the mechanism behind this kind of skewed phenotype that results in the exaggerated chronic mucocutaneous candidiasis picture. So basically, because of the mutations being where they are, you're having an impairment in terms of unactivating or deactivating the STAT1 protein. And again, the resulting clinical phenotype, you see chronic mucocutaneous candidiasis can be oral, can involve the nails, can involve certain skin fold areas. So something that everyone should think about if they have a patient that comes in with recurrent candidal infections. This is a possibility, something that they could have as part of their underlying reason for their disease. It would be a STAT1 gain of function mutation.
I think what's worth stressing there too is, while all of these other diseases are extremely rare, STAT1 gain of function actually appears to be extremely common relative to all other, immunodeficiency
That's a good point.
The phenotype is pretty broad.
Mm-hmm.
Does it have any other, is that the worst that it causes, is chronic?
No, it
There's
causes
other
far
things,
worse. It can, right? It can be very mild. There's also a predisposition to viral infections, especially herpes viruses in some people. Some people will have an antibody deficiency associated with it, and it's not fully clear.
You can have aneurysms associated with that picture as well. So there's a few different things that seem to be associated with the STAT1.
Talking about it as if it's one defect, but since it's a complex protein, can you have different people have different genotypic abnormalities and hence more severe phenotypic expression?
Yeah, there hasn't really been, nobody's correlated that, there have been 20 mutations that have been described that lead to gain of function, and nobody's been able to correlate the mutation with the phenotype. So it doesn't appear that simple, which is common with genetic mutations.
So moving on to STAT2. STAT2, again, is important for signaling for interferon alpha and beta. It is involved with, this pathway goes through JAK1, tyrosine kinase 2, and then leads to STAT1 and STAT2 activation and phosphorylation. So these two are fairly closely linked. We talked about STAT1 deficiencies and how that can lead to a more severe immunodeficiency with pretty bad susceptibility to viral and mycobacterial infections, whereas with STAT2 deficiency, there's been one family described where there was kind of a varying degree of phenotypes, and it seemed overall more mild than what you might expect from someone who has a STAT1 deficiency. So again, this schematic just shows you how STAT1 and STAT2 are associated in terms of interferon alpha beta signaling. You see STAT1 and STAT2 dimerize with each other and go into the nucleus and affect transcription. So obviously STAT1 deficiency by itself may cause a different type of clinical picture, but interestingly, when you have just a STAT2 deficiency, it doesn't seem to be as severe as STAT1 deficiency by itself. So what this paper showed was that there was one family that had 5 patients that were affected. You can see in the pedigree here the patients that were affected. There was one patient who had a fatal viral infection that was an infant. They died a couple of days after birth with what was presumed to be a viral infection. And there was also The proband was actually a patient that developed vaccine-strain measles. They got a measles infection from the vaccine, and then subsequent to that, they realized there had been a child that died in infancy from what they thought was a viral infection, so they looked into the family in more detail. And what they found was the other 3 affected children, aside from the one who died in infancy, all had some degree of viral infection that at one point required hospitalization. There was also a second child that had some kind of encephalitis type picture after having received MMR. But they were all otherwise relatively normal, and there was even an adult who had STAT2 deficiency by genetic findings, but appeared to be relatively asymptomatic, at least in adulthood. There wasn't much history about whether there had been any significant viral infections for that adult as a child. But basically what they were showing is there's seemed to be a varying degree of phenotypes within this one family for those who had the STAT2 mutation. So it did seem to be a predisposition to having more severe viral illnesses or perhaps issues after having received live viral vaccines, but they didn't seem to be deadly or lethal, aside from the one who died at 2 days of age. Again, comparing and contrasting this to STAT1 deficiency, where generally the phenotype is much more severe. They mentioned there was one patient in here who had primary HSV, and their presentation was kind of a gingivostomatitis picture, whereas they compared that to patients with STAT1 deficiency that generally, if they're infected with HSV, will go on to have encephalitis in a much more severe picture. So appears to be similar but less severe with regards to comparing STAT1 and STAT2 deficiencies.
So does STAT2 only form a heterodimer with STAT1? It doesn't form a dimer on it?
That was what I understood. STAT3, you guys are probably familiar with the dominant, autosomal dominant form of hyper IgE syndrome, which is caused by STAT3 mutation. Basically, you get mutations in the DNA binding and SH2 domains, and this causes a different type of phenotype, which shows things such as eczema, recurrent skin infections, recurrent pneumonias, particularly cystic pneumonias, and issues with hematopoiesis. They can also have other types of abscesses. They can have chronic candidiasis as well, and they generally have very, very high IgE levels. In past studies, it's been shown that this mutation is correlated with decreased levels of Th17 cells in peripheral blood. Some people have kind of suggested that could be a marker. There's obviously some issues with trying to decide, is this individual just atopic and very, very atopic and unlucky, or do they have hyper IgE syndrome? And obviously, by doing sequencing of STAT3, you can figure out if a STAT3 mutation is the reason for it, but there's interest in trying to find other ways to perhaps diagnose this without having to do the sequencing. And so people suggested that looking at Th17 cells is one way. Autosomal dominant hyper IgE syndrome has kind of a characteristic phenotype. You can have hyper IgE syndrome due to an autosomal recessive mutation as well, which also has things such as high IgE, the eczema, and skin findings, but there's other features that are more typical of the autosomal dominant form, and those include things such as kind of typical facial features, issues with hypermobile joints, and bony abnormalities as well. This picture just kind of highlights the prominent forehead and the wide nasal bridge and other features that are prominent in this family where both a parent and child were affected. This down here is just showing kind of the cystic pneumonias and abscesses that can form in these patients as well.
At the bottom of that, it has NIH score over a certain amount of points. What is that? What is NIH?
Yeah, so basically there's a scoring system where you look at these different features and you get certain points for how high their IgE level is, if there was presence of a rash as a newborn or not, what's the most severe form of eczema they've had, have they had issues with fractures, have they had issues with recurrent candidiasis, how many times, and each certain thing gives you a number of scores. If you get a score between 20 and 40, that's kind of an intermediate range saying, yes, it's possible this is the cause of the disease. You may want to consider sequencing. If the score is 40 or greater, then they figure that is a pretty sure bet that they have this disease, and in that case, you should probably...
It's specific for this disease?
For the autosomal
Okay,
dominant
does it
form.
have an NIH score for all different diseases?
No,
Like
just for the autosomal dominant form. They have a scoring system, which you can look up, and if you're considering, is this just a really atopic individual, or do they maybe have hyper IgE syndrome, that scoring system may help you decide if it's worth looking into sequencing STAT3 or not.
Is the second column there people with
So these are people
clinically confirmed hypereosinophilic
hyper IgE syndrome, but without the STAT mutation. Exactly. So as you can DOC8 deficiency is proposed to be a reason for autosomal recessive forms of the disease. And again, in those individuals, what you see mostly is the really high IgE and the eczema. Those are very common features in both diseases. But some of the other findings, as I mentioned further down the list here, are less common in the other forms of the disease when compared to the autosomal dominant form, which is caused by the STAT3 mutation.
What do they think the causes of the second group?
So as I was saying, the autosomal recessive form has been linked to DOC8 deficiency.
So that's a DOC8 deficiency, is it?
What's that?
TIC2 also.
Yeah, and right, and I'll right. So we talked about that a little bit earlier. The tyrosine kinase. There's been a couple different mutations described in one individual. In that case, also had kind of a hyper IgE syndrome picture. The one who had kind of that accelerated Th2 response had increased levels of IL-5 and IL-13 when stimulating the CD4 cells. So they were proposing that someone with a tyrosine kinase 2 deficiency could also fall into that category of someone who's not autosomal dominant and might have another reason for having had hyper IgE syndrome. So that's been another proposed mechanism.
Is there a spectrum between these two columns in terms of level of IgE? In other words, does the left column tend to have higher levels?
That's not necessarily clear, and I'll go into more detail in a couple of the other studies that I looked at when they're kind of trying to decide, is this individual atopic or do they have hyper IgE syndrome? And in those studies, they've shown that both people who just have really bad atopy versus people who have hyper IgE syndrome have similar levels in terms of the elevated IgE. So you can't really say, well, this is a really high IgE, so they are more likely to have autosomal dominant hyper IgE syndrome. Yeah.
Is it non-immunologic abnormalities like the facial features and so forth? Is it understood how STAT3 is involved in the embryogenesis of those tissues?
I'm not sure, honestly. I don't know if either of you guys have any thoughts on that. Yeah, I'm not sure the reason for that. This was a paper that was looking at other features of autosomal dominant hyper IgE syndrome. This was from a French study. And one thing that they noted is that, yes, the patients had higher IgE levels in general than what would be the norm, but they also found that they had lower levels of CD27 positive B cells. And they proposed that this could be another feature that may be unique to autosomal dominant hyper IgE syndrome and may be easier to screen and evaluate for than the Th17 cells. As the Th17 cells, that requires looking at a smaller population since the levels reduced in these patients, and it requires intercellular staining due to the presence of such a small T cell population. Therefore, they were suggesting from this paper perhaps looking at the presence of the CD27 positive B cells and showing that those were much reduced compared to a norm would be another way to diagnose this without having to do STAT3 sequencing. Of course, this is just a proposition and not exactly what everyone's doing at this point, but interesting finding nonetheless.
So Hans and Troy published a paper a few years ago looking at this, and they compared the NIH score and looked at Th17 cells. I think Troy does that assay still, correct? And if you look at both of those things, it's fairly accurate at predicting whether people have either the STAT3 or the DOC8 mutation. So doing the NIH score and getting that, I've done that several times through Troy's lab, and it's been fairly predictive. I haven't sequenced anyone because it was so low likelihood on both accounts, but they published, I think in 2012, they have a fairly accurate predictor of that.
I think the sequencing is more reliable than Th17. How do they do that anymore? When you did the Th17 study on STAT3 patient, they basically have zero Th17.
Right.
STAT1 gain of function about 50% every time, but still, sequencing is more reliable.
But the point was that the score alone is certainly adequate to rule out STAT.
Right.
Score alone is reliable, and I think they published another paper comparing DOC8 to STAT3, and again, they validated the power of the scoring system. So you can also differentiate between DOC8 and STAT3.
Lisa, what's the CD27 code for?
So those are types of memory B cells.
Okay.
What's the little bar up there? Is that just
And
plotting the different genetic defects?
This
No, the
this
other
appear?
one up
It's
here.
negative.
Yeah, so this appears showing kind of where the majority of the identified STAT3 mutations are, and they seem to center around the SH2 domain in the DNA binding region, the ones that have been identified so far. This is another paper talking about STAT3 mutations and how they were able to determine that this actually caused a change in mast cell degranulation. So again, this was comparing a bunch of allergic people with people with STAT3 mutations causing hyper IgE syndrome. So what you see is that there were similar levels of eczema among those that were atopic versus having hyper IgE. There were similar levels of elevated IgE. However, those with STAT3 mutations causing hyper IgE syndrome actually seem to have lower percentages of physician-diagnosed food allergy and physician-diagnosed anaphylaxis. So basically, this paper was commenting that when you see a patient who has very high IgE levels and has a pretty severe form of atopic dermatitis, generally those who have the autosomal dominant form of hyper IgE syndrome actually have less other allergic disease, whereas you might expect that a very atopic patient is going to have high IgE, they're going to have severe atopic dermatitis, but they're also going to have food allergies and asthma and allergic rhinitis and a bunch of other signs of atopy, whereas that's not what you see with these patients with STAT3 mutations. And so they were proposing different mechanisms for why this might be the case. And what they found was that they seem to have compromised mast cell degranulation. They did different levels of morphine as a skin prick test to see if the patients formed a wheal and reacted to it. And what they found was the proportion of patients having negative skin tests in the STAT3 mutation population was higher compared to the controls, even at higher levels of morphine. So these patients were not able to form a wheal in response to the morphine skin prick test, even though their histamine wheals were relatively the same. They also looked at basophil activation, and basically they looked at IgE cross-linking as a marker of this, and comparing the volunteers to the STAT3 mutation population, they were showing that there was less activation of basophils, meaning less cross-linking of IgE when stimulated, when you looked at the healthy controls compared to those who had STAT3 mutations. So they're proposing that impaired mast cell degranulation and impaired basophil activation is one of the reasons why you see less allergic types of diseases in these patients with STAT3 mutations. Okay, and then I'm also going to talk about STAT5B mutations as another type of kind of unique clinical phenotype you can see, and something that should be considered if you come across a patient with some of these features. So STAT5B, this is involved in the signaling pathways of things like IL-2, IL-4, different colony-stimulating factors, and growth hormone, if you remember, I mentioned that earlier. What you see with STAT5B in terms of the immune system is decreased numbers of CD4, CD25 positive T cells, so regulatory T cells. They also find that patients with these issues, patients who have autosomal recessive mutations in STAT5B, have a picture that not only results in immune deficiency because of differences with regards to the regulatory T cells, but also autoimmunity, growth failure, and some other clinical features that are relatively specific. So again, this is just showing STAT5 signaling, the different cytokines that we mentioned that are involved, particularly growth hormone and IL-2 signaling through the STAT5 pathway. This leads to issues, or not issues, but leads to things such as activating T cells. It normally leads to expression of FOXP3, which is important for Tregs, and also leads to growth through expressing IGF-1. There's been several different mutations that have been identified in various portions of the gene so far. And the things that you see in these patients is dwarfism, which again is resulting from the issues with IGF-1 production leading to poor growth. They also have some prominent features such as large foreheads and saddle noses. The issues with the regulatory T cells results in low NK and T cell numbers. And so these patients will have recurrent viral infections and other opportunistic infections, specifically PCP is seen frequently. They also have other features. They have things like eczema and kind of a failure to thrive picture with early onset diarrhea. But one of the most prominent features you see in these patients is also a severe interstitial lung disease. So the lung disease is generally the larger source of morbidity and mortality in these patients. One thing that was mentioned with regards to that when I was reading about this is that some studies suggest that patients with common variable immune deficiency that have lung disease as well, that have interstitial lung disease, show decreased levels of phosphorylated STAT5B. And so they're proposing perhaps there's some mutations in that pathway that might underlie individuals having issues with lung disease who have CVID. As we all know, there's no common genetic mutation for CVID patients, and so it's interesting to think about which specific genes, perhaps in the JAK-STAT pathway, some of them may be underlying the different phenotypes you see in patients with CVID. And then again, with STAT5B deficiency, this is a family where there are two siblings affected. It's difficult to see because you're not seeing a normal person next to them, but they are relatively short and have impaired growth. The older sibling is also on oxygen, as you can see, secondary to severe lung disease, and they do have some of these kind of, you can't see as much, but prominent foreheads and nose deformities, as was mentioned. This is showing growth from a couple of patients that were noted in one particular paper that were found to have STAT5B deficiency, and again, we see that they have linear but much decreased growth and compared to what is normal for boys and girls of those ages. And again, these patients, their growth hormone levels are actually normal. They're insensitive to growth hormone. They're just not able to produce IGF-1, and so they have very low IGF-1 levels. So growth hormone itself and trying to give growth hormone to these patients as a reason or as a way to try and catch up their growth is not effective. They need the IGF-1. So this was something that was brought up. It was published in Pediatrics a while back, stating, if you have a child with failure to thrive, with a poor growth type picture, think about this as a possible mechanism for the disease, particularly if they're having issues with lung infections, to try and prevent them from having so many lung infections before you catch it to the point that they're going to have more severe disease from it. But I thought that was somewhat interesting with regards to this. So in conclusion, the JAK-STAT pathways obviously have a wide variety of roles in the control of immune responses. In dysregulation of this pathway is associated with a wide variety of different types of immunodeficiencies, everything ranging from SCID to maybe just predisposition for some viral infections that may or may not be severe, depending on if you have a STAT1 or STAT2 deficiency. We talked about the gain of function mutations with STAT1 as well that can provide a pretty unique phenotype with recurrent candidal infections, as well as predisposition to viral infections and other things. And then the STAT3 and STAT5B mutations have kind of their own unique phenotypes as well, with the hyper IgE syndrome and then this one that's associated with immune system issues, lung disease, and dwarfism with the STAT5B, as I've mentioned. So they do have varying roles. They have impacts on a bunch of different genes and lead to pretty varied phenotypes depending on what mutation you're looking at. And then, as I mentioned just briefly, mutations in this pathway, gain of function mutations generally, can also cause issues with autoimmunity, cancers, myeloproliferative disorders, which makes them a very attractive target for inhibitor therapies such as inhibitors of JAKs. But unfortunately, that's not something that's going to help with these loss of function mutations that are leading to deficiencies, which is mostly what I spoke to you about today. Yes.
Are any or all of these treatable with bone marrow transplant?
Some are.
It'd be a real bummer to find these people in clinic and say you have this pathway, you're going to die. And
right.
Is there an option in therapy there?
Right. Well, I mean,
just a bummer. Oh.
Yeah. I think it depends on, obviously for things like the JAK3 mutation where they have a SCID phenotype, then yes, bone marrow transplant is considered. For some of these other phenotypes where there's kind of or these immunodeficiencies where there's a range of phenotypes ranging from mild to severe, it may be more of a case-by-case decision about whether or not that, if it's someone who's just having multiple severe infections recurrently and they could perhaps have a good quality of life following transplant, it's something to consider, I suppose. But it's not routinely done for every
Theoretically,
mutation.
all these could be corrected with a good bone marrow transplant.
Well, only the immune aspects of it, because when you have vascular disease and other growth issues, so I think with STAT5B there have been some transplants, and the question is then in terms of the growth,
Won't be affected.
It won't be affected, and so these, although we focus on the immune system here, function in every cell in the body, and
It doesn't correct everything, just immune
except the
defect.
immune, and really the hematopoiesis. So also.
Some cleavage is pretty much a bummer.
No, they won't die of infection.
Lisa,
Yes.
do the drugs cyclosporin and tacrolimus interfere in other areas besides interleukin-2? Do you know in different pathways?
That's a good question. I'm not really sure.
Whether the calcineurin inhibitors affect signal through this line too.
Do they affect the function of these proteins? Is that what you're asking?
No, in terms of
Other
therapeutic
pathways,
effect.
yeah.
Is it also through some of these pathways?
Yeah.
Just wondering.
Yeah.
For the future, what will be a cost-effective immunodiagnostics for this? How will people be screening? When we're suspecting these, what would be the?
Yeah, I mean that's a good question. I think some of these, are more straightforward than others. We talked a bit about the STAT3 mutations and how there might be other ways to screen, such as looking at the Th17 cell population or looking for CD27 positive B cells and seeing low levels of those being a marker for the disease rather than having to sequence. But I think for some of them, unfortunately, sequencing is the way to do it at this point. I think maybe as more and more mutations are identified, it might be easier to say, this specific phenotype goes with this disease, and you might not have to sequence to say this is a STAT5B deficiency. But I don't know that we're there yet with a bunch of the mutations I described. There's just a few, a handful of cases that have been talked about here and there. So.
With the cost of sequencing, all the focus now is just on doing that versus functional assays. Because if it's a clear phenotype, you sequence STAT3. It's $1,000. If it's unclear, so you're going to be able to sequence all of these.
I mean, I believe that you need a functional assay. We just had a patient with STAT1 gain of function mutation several weeks ago, and he had a second mutation downstream to that mutation. So when you do a functional assay, checking phosphorylation in response to different stimulation, he didn't react like STAT1 gain of function. It was STAT3 basically. Like the second mutation abolished the function of the gain of function. So you need to have the functional assay to evaluate the response. And I think another point that probably we should mention is there is a crosstalk between, or some balance between all the stats. So every stat basically inhibits other stats. So in the case of Th1, Th2 development, once you get STAT6 phosphorylation, then it inhibits STAT4 and you induce Th2 instead of Th1. It's the same interplay between STAT1 and STAT3. If you check the level of STAT3 in patients with STAT1 gain of function, basically they have almost zero level of STAT3. And if you check STAT3 autosomal dominant hyper IgE patients, they are starting with very high level of STAT1 phosphorylation. So that's another reason why they have the chronic mucocutaneous candidiasis. It's only that maybe STAT3 is important to Th17 production, but the fact that they also have elevated level of STAT1, that's another reason why they suppress Th17. So there's always an interplay between all the stats.
Yeah.
How many times is this gene coded? I mean, if you look at for the JAK proteins and the STAT proteins, are they all in one chromosome, or are they spread around the genome in multiple places?
The different JAKs and STATs?
Yeah. Are they will one mutation take it out, or is there more than one copy?
There have to be two copies.
Yeah, there's two copies.
Right, but I mean, in one chromosome, are there more than one location on a chromosome?
I think they're spread all around.
Yeah, I don't know all the different chromosomes they're located on, but they're not all on the same chromosome.
David? Aren't they
I don't
all
think
different chromosomes?
they're not on the same chromosome. I think there's only two copies of each. So they don't have it's not the reason. It's a copy number variant.
There's a question from the outside here.
Yes.
Slide 37, so someone was actually listening.
Wow.
Shows STAT5 resulting from activation of receptors for growth hormone, IL-2, 9, 13, 15, 21, EPO, BCSF, and TPO. How does the nucleus of the cell that received dimerized STAT5 know which ligand sent the signal that was transduced? That's a very elegant question.
Yeah.
Chuck sent that.
Sorry, so the question was, how does STAT5 know which signal, which cytokine
That's the
is
question
signaled?
I asked earlier. How does the biologic system know all the crosstalk, all the way from stimulus to DNA, to know what, how to translate the signal?
Yeah, that's a good question. I don't know all the intricacies.
It's the same. It's the combination of the receptor and some changes in the DNA which allow the STAT to bind to specific sites. So different cells, the different chromatin and the binding sites on the DNA themselves are changing between different cells.
Well,
so
all
you have
cells
different
have the
receptors.
same chromatin. They just don't activate it all.
So first of all, you have different receptors. So you won't have the growth hormone receptor on every cell. And you won't have the erythropoietin receptor in every cell. So to begin with, you can't respond. Not every cell can respond to the same stimulation. And the second phase will be some genetic changes in the DNA itself. So STAT5 will be phosphorylated. It won't bind to the same place in each cell. So different cells will have different binding sites.
So
I guess it's a combination
answer
of two.
for those, the specificity in the system, each cell figures it out for itself in a very simplistic statement.
But there'd be spatial differences too from what, like a dendritic cell in the airways would be producing a different mix of cytokines locally, depending on what local cells are responding to it. So you'd have your architectural differences there. And then there'd be quantitative differences also from where let's say a dendritic cell is producing it, what kind of, how robust the production of the cytokines. And then you've got your cells that are coming back in too, the inflammatory cells infiltrating, they're responding different ways depending on their state of activation. So,
But I think the main point is only certain cells recognize certain cytokine signals to start the process in the first place. They don't. Only certain cells will see growth hormone, other cells will only see IL-2 and so forth.
It's a kind of basic question, how a single cell develops into two different cells.
Here's a very trivial question compared to these elegant questions. Why is JAK4 called tyrosine kinase 2?
Right. So, from what I read, that was one of the first ones described, and it was a tyrosine kinase. I'm not sure why it was 2, but they called it tyrosine kinase 2. And then after that, when they described all these other ones, those were described as Janus kinases. They realized they were all the same group, and so it's part of the JAK family, even though it was initially labeled as just a tyrosine kinase. Yeah.
It's like complement cascade is C142 because they discovered it out of order.
Right. That happens.
Something like that.
Any other questions that I can try to answer?
I think we can officially say the fellows are educating us now.
Yeah,
absolutely. The torch has been passed.
Thank you.
Next week, Gary's our speaker.
A little simpler.
No, you Big Bang Theory that you did, that wasn't simple.